In this video, we're going to look at the problem that cells have at replicating the ends of linear chromosomes. And it's called the 3' end problem. So if we take a look at the top image here, we're looking at the complete end of a chromosome. And the dark blue, those are the parental strands, and the light blue, those are the new strands that are being made. So we're replicating this chromosome here. Now, if you take a look at the leading strand that's going, that's following that replication fork to the end, that's not a problem. That DNA polymerase will fall right off the end of the chromosome and be able to replicate that particular daughter strand, no problem. But look at this, the lagging strand. So we're looking at, essentially, basically, what we're looking at is a replication fork and taking it all the way to the end of the chromosome. So with every replication fork, we've got that leading strand, no problem there. But look at the lagging strand. Right here, let's look at this boxed area and magnify it. And what we see is that last Okazaki fragment puts down that last RNA primer. Well, the problem we have here is that our DNA polymerase 1 that usually removes that primer and adds nucleotides, DNA nucleotides back, it needs a primer. It itself needs a primer, and yet there is no primer. There's nothing at the end here. So that DNA polymerase 1 cannot fill in DNA here, because there's no previously laid Okazaki fragment that it can use as a primer to fill in this area with DNA. Which means every time a cell divides, every time DNA replication occurs to a chromosome, that chromosome is going to shrink by about the size of a primer from both ends of that chromosome. Now, the solution that the cell has, okay, is more of a workaround and not a solution. And that workaround does not stop chromosomes from shrinking every time the cell divides. Instead, the way the workaround works is early in your embryonic history, right? When you are just an embryo of, say, hundreds of cells or so, your cells express a protein called telomerase. And telomerase is a ribonucleoprotein. It's made up of a protein and it's made up of RNA. Now, that RNA is reverse complementary to these what are called telomeric repeat sequences that are at the end of every chromosome. And so what happens is that telomerase is going to act as its own template to add more of these repeated sequences to the ends of all of your chromosomes. So we've got an AAUCC here in the telomerase. That's going to bind to a TTAG, and then the overhang here is going to be used, and it's going to catalyze the production of some more DNA. A C to a G, a C to a G, an A to a T, an A to a T, a U to an A, and a C to a G. So it's going to make this repeat. Now, that telomerase shifts up, shifts over, shifts back down, and look at that. And now it can make another repeat. So these telomerases are going to make hundreds of these telomeric repeats, these repeated sequences at the end of all of the chromosomes. But that only happens during embryonic development. Eventually that telomerase shuts down, and the size of your telomeres on the ends of all your chromosomes, that's it. That's as long as they're going to get. Now let me mention one special thing about telomerase. First of all, notice it is a polymerase, right? It can make DNA. But what is it reading? It's reading RNA and it's writing DNA. So telomerase is an example of a reverse transcriptase. Reverse in that instead of reading DNA to make RNA, it's reading RNA to make DNA. So it's kind of a neat enzyme. If you were to take your chromosomes and if you were to stain them with a chemical that binds specifically to the telomeric repeats, you would see that all the ends of all of your chromosomes have these telomeric repeats. What's special about them? Well, what's special is that they do not encode any genes. They don't really encode anything important. So now, when we're looking at that last Okazaki fragment with the last primer in green there, we're still going to lose some material here. We're still going to lose some DNA, but the DNA that we're losing is just telomeric repeats. Okay? But remember that telomerase is no longer expressed in your cells after embryonic development. The number of telomeric repeats, the length of your telomeric repeats at the end of your chromosomes that's it, maxes out. And then every time your cells divide, your chromosomes are going to get shorter and shorter and shorter. Now, sure enough, that's going to just eat through your telomeres. No worries, eat through your telomeres, but there's going to be a point at which your cells go, whoa, 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 wait a minute. We can't divide anymore because we're running out of telomeres. And those cells stop dividing. That's called the Hayflick limit. If you take most of your somatic cells and you plate them out on a plate and force them to divide, force those cells to divide, they'll reach about 20 cell divisions and stop. It's about that many cell divisions where the telomeres of the chromosomes get too short and the cells say, no, if we divide any more, our chromosomes are going to be so short that we're going to start messing genes up. And so they hit that limit and they stop dividing. Now, along with that comes tissue damage, tissue degradation, and this is what we think is involved in aging. That one of the reasons that you go through all these various phenotypes of aging is because your cells reach this Hayflick limit, your telomeres get too short, and you can no longer sort of regenerate any of your tissues, and you then have these aging phenotypes. But let's think about that. If that's really true, if the length of your telomeres and the fact that they keep getting shorter and shorter and shorter every time your cells divide, if that's what's causing the phenotypes of aging, then it would be a decent hypothesis to say, well, what happens if we force cells to express telomerase, right? Normally, telomerase is done. After your embryonic development, it's turned off, never turned on again. What if we were able to turn them on again? Well, there's some experiments that have been done to test that. There was a line of mice engineered that they had a way of selectively turning off the telomerase of these mice. And so for several generations, they kept the telomerase off, even during embryonic development. And so by the third or so generation, mice were being born that had short telomeres, and within just a few weeks, 30 to 35 weeks or so, they were already showing symptoms of aging, short, dysfunctional telomeres, increased DNA damage signaling, systemic degenerative pathologies, that's the tissue degradation. So they were starting to show these premature aging effects because of the small telomeres. And at that point, when they started showing these phenotypes of aging, they then turned the telomerase on. So they turned it on, and what did they see? Well, the telomeres started to extend again. Well, that was expected. But then it reduced the DNA damage signaling, right? The cells were no longer saying, hey, hold on, we got to stop dividing because our telomeres are too short. So no more DNA damage signaling, and then most interestingly, a reversal of those degenerative pathologies. So a reversal of some of the tissue degradation that was occurring because of the aging. So this is good evidence that in some cases, or at least in some respect, the length of your telomeres determine when you are going to start showing these age-related pathologies. And there's one company and one woman, Liz Parrish. She's the CEO of BioViva Sciences. And gosh, I don't know how many years ago it was, but she did something that is illegal in the United States, and she went to another country to do it. Her company was developing a system where she could force telomerase to be turned back on in a bunch of different cell types of hers. Okay, it's called gene therapy. And she was her own patient zero. She injected herself with these viruses that were engineered to turn telomerase back on in her cells. The idea being that it increases the length of her telomeres, and she did this for her T lymphocytes. And sure enough, within 6 months, her telomeres went from 6.71 kilobases to 7.3 kilobases. Now, unfortunately, right, she works for a private company. Well, she owns a private company. They are by no means required to publish the progress of how she's faring. She is still alive. She hasn't gotten cancer because, right, that's one of the possibilities. Is one of the key features of a cancer cell is that it can just keep dividing, keep dividing, keep dividing, keep dividing. Well, how can it do that? How does it break the Hayflick limit? Well, one of the mutations that occurs to those cancer cells is that their telomerase is turned back on. So the idea that telomerase is usually turned off in our cells is probably a protection against cancer. So that's one of the worries about, oh, let's just turn telomerase all on back in our cells. The worry is that you end up with getting cancer. Well, she hasn't gotten cancer yet. And I don't know the extent of her telomeres and the extent of what sort of reversal, any pathologies. So this is something we can look out for. And I've noticed that she has more and more videos online on YouTube, so you can research her that way. All right, well, DNA replication is absolutely essential, right? Because it is going to be all of the instructions for that new cell, right? Because DNA replication occurs when a cell divides, for that new cell to function properly. And so if you replicate DNA incorrectly, then you are going to have a incorrect set of instructions, and that cell may become cancerous or something like that, right? So that damage that might occur during DNA replication or any other damage has to be fixed before it becomes a mutation. So DNA damage or a lesion can be detected. It's something abnormal that a cell has a system in place to detect. Mutations, once a lesion becomes a mutation, a cell can no longer fix it. So let's look at this example. So we have a normal double-stranded DNA, and now it's going to divide because the cell is going to divide. When this top strand here is a template for the production of this bottom strand here in this one cell, everything is normal. C to a G, C to a G, T to an A, T to an A to a T, G to a C, everything's normal. But look what happens in this cell. We have our parent strand right here, but where you should have an A to this T, there was a mismatch. A G was put in there. That's a mistake that can sometimes happen during DNA replication. It's called a mismatch. But a mismatch has an altered shape that the cell can recognize as wrong, and so it is a lesion. It's not a mutation yet. Okay, now, if that lesion is not fixed at this particular point, then this is what's going to happen. So this parent strand is going to serve as a template to make this, and that's going to be normal, right? This one is completely the same as this grandmother DNA here. But this one here, if that G doesn't get fixed, and it's going to be now the template, and if it gets read right, that G to this C, boom, this is now a mutation. We used to have an AT base pair, and now it's a GC base pair, and there's nothing that cell can do to detect that should not be the way it is. It is now a mutation. So what we need to do is let's figure out what kind of systems does the cell have in place here to detect and fix lesions, damage, before they become mutations. First, let's take a look at sources of these lesions. The biggest one are these mismatches during DNA replication. There's also spontaneous chemical damage, where, for example, cytosine can become uracil, and they'll pair differently. There's chemical damage like UV light, deaminating agents, oxidative damage. This is a thymine dimer. This occurs when your cells absorb too much UV irradiation from the sun. And a thymine dimer is going to cause the DNA to kind of bend, and that's going to cause problems during DNA replication. Now, how can we fix these lesions? Well, DNA polymerase has a limited proofreading key, so it could add something, and then there's a little bit of time that it can detect, wait, that was wrong, go back, delete it, and add it again. But that's only like one base pair at a time. It does increase DNA polymerase's accuracy by quite a bit. In addition, there are a ton of repair systems within our cells. There's all sorts of genes. About 130 genes in humans have evolved that are involved in DNA repair. Okay, and here the loss of the function of any of these DNA repair genes are going to lead to the development of cancer. Why is that? Why would a mutation or a defect in your DNA repair system lead to cancer? Well, if you can't repair your DNA, you're going to get a ton more mutations, and now the chances of that one of those mutations hitting a cancer gene goes up. So it's just a numbers game. There's lots of different repair systems that repair different things. Let's take a look at the nucleotide excision repair system. And what this does is it's a system that recognizes these thymine dimers. So here's that thymine dimer, the two thymines that have been covalently bonded and giving this abnormal structure. Well, what all of these systems have in common is they have a protein that recognizes this specific lesion. And when they recognize it, they bind it. And instead of directly fixing it, what they more often do is they cut, and just remove all of that and eat up all of that single-stranded DNA. And then just replace it. So they cut, remove, and then we have our DNA polymerase, right? And this is a special DNA polymerase. It's more like DNA polymerase 1 rather than DNA polymerase 3. Now, and notice it doesn't need a primer because it's using the existing strand. Just like DNA polymerase 1 uses those upstream Okazaki fragments as primers. And then it adds, but remember it can't add one existing strand to another existing strand, so what do we need? We need DNA ligase. And many of these repair systems work this way. A protein that recognizes the damage and eats the DNA around it, a DNA polymerase that fills it in, and a ligase to fill that nick. All right, so now that we understand how DNA replication works, we're going to look at the entire mitosis and cytokinesis and the cell cycle. How does a cell divide and how is it regulated, that cell division regulated?